Alright, this is another video. This is the unit that we are doing about DNA and biotechnology. So if I won't give the unit name because it could be different by the time some of you are viewing this, but it is about DNA, and thing you might have, the pages you might have in your unit might look like this. If not, these are a lot of review questions, and I know sometimes when I've made these videos, I've given you a few places to stop or notes to take, or I've given you something the next day to check, to see if how well you've done. This is a good one, this slide presentation, because even though there's a lot of slides, a lot of them are questions that you just pause and answer it, and then I'll move on. So we'll go over those in class. That would be a really good thing to do, so that way I don't have to lecture you on all of this in class. So we're going to talk about DNA today. We've really been talking about it quite a bit, if in the flow of things, for the whole year. But let's talk a little bit about what is DNA. It is called deoxyribonucleic acid. The DNA stands for that. Deoxyribo is a nucleic acid. It is your instruction manual for your life, and it's in the nucleus of your cells, and it is the chemical or molecular basis for how we pass on our genes. It's the chemical of life. You probably know this part. It's found in the nucleus, as I said, and most of the time it's very loosely like strung in the nucleus, and you can't even tell the chromosomes are really even there. And if you are in a particular place in the unit, you've probably actually even looked at cells with the nucleus. Then at that point, it's called chromatin. TIN at the end. That is basically the same thing as chromosomes, but they're loosely kind of arranged. They're not thickened up like they do in prophase that we talked about. And as you probably remember, I've discussed that a gene is what makes up a chromosome. Genes are sections of a chromosome that code for specific traits. So strands of DNA make up genes, and genes are what make up chromosomes. So DNA makes genes. Genes are made of this molecule called DNA, and when you put genes together into these structures, they're called chromosomes. And when the chromosomes are loosely coiled in the nucleus, they're called chromatin. You can replay that if you want. So this is question number 1. This says the loose, thin fibers of DNA and protein are called blank. Choose from one of those 3. Now, we also refer to what DNA is as a polymer. A polymer, poly means many, and so it's made up of many of this repeating unit, which is called a nucleotide. A nucleotide consists of a sugar, which is called deoxyribose. Remember, all sugars end in ose. Something called a phosphate, which is the same thing you saw with ATP. If you are in a class where we studied ATP or chemical energy, it's a phosphorus with 4 oxygens. And then it's got one of 4 different bases on it. I don't even really do the names too carefully, but A, G, C, and T, adenine, guanine, cytosine, thymine. So the 3 parts again are sugar, phosphate, and a base. And that's repeated over and over and over again, so we call it a polymer. And that's what DNA is. It is double-stranded, which basically means it's kind of like a ladder. It has a side with a railing and another side with a railing and steps in the middle. That's called being double-stranded. And then they're wound together to make kind of a spiral structure, so that we call that a double helix. Helix means twisted, so like a twisted ladder. And that's what it kind of looks like. I'm not going to say too much more because I'm going to ask you some review questions, but hopefully you'll do some reading in your book about this too. But that's a picture of DNA with the rungs or the steps of the ladder kind of being in the middle with color, and the sides of the railing of the ladder or the staircase, those are the kind of bluish gray things there. So I guess I should probably point those out. So this part right here is the sides, and then these are the bases. So it's like a ladder or staircase that's twisted. So what is the what? Well, the backbone, or the sides of the ladder, the parts where you'd put your hands to climb up, that's made up of a repeating sugar, and then phosphate, then sugar, then phosphate, then sugar, then phosphate. And connected to the sugar is one of the bases, A, G, C, or T. And then you have another side of the DNA molecule that's repeated on the other side, so it ends up looking like a ladder, as opposed to a half of a ladder. So take a look at that picture there, and take a guess. Which of the choices, 1, 2, 3, 4, and 5, which one represents the sugar-phosphate backbone, or the sides of the ladder or staircase? And again, if you need to go back, you can always go back. Now I would like in the video or in your book, now I'd like you to figure out which color represents the bases. 1, 2, 3, 4, or all of the above. And here is a picture of a nucleotide. Okay, it's a sugar. It's this ring structure here with a base, in this case G, with that phosphate. There's the PO4 right here. Here's another one. Here's another one. Here's another one. Remember, there's 4 different kinds. One with G, one with C, one with T, and one with A. And all the sugars and phosphates are exactly the same. So this makes up the side of the ladder. And this makes up the middle part of the ladder. And then there's another side, so you get a full ladder. You'll see that. It ends up looking something like this. And again, this is sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate. And connected to the sugar, to this pinkish structure, these are the bases here in the middle. So it ends up looking kind of like, again, a twisted ladder, a double helix, a spiral staircase. We have the name for bases. I told you that it's T, C, A, and G. Well, T and C are kind of like in the same family, and they're called pyrimidines. And A and G are purines. And the reason that's important is because there's a kind of a rule that we've found out over the last 50 years or so, and that is that same families don't like to hook up with each other. That's kind of gross. So T will always match with A, one from each family, and C always matches with G. And the way that you can tell them apart is based on whether they have 1 ring or 2 rings in their chemical structure. Now, try this one. How many bases are there? How many different kinds of bases are there? How many different kinds of bases are there? So, let's go back and go to previous, and let's go to previous again. So the first question says, how many different bases are there? 1, 2, 3, or 4. This one's saying, how many different groups or kinds of bases are there? 1, 2, 3, or 4. So as I mentioned, a 1 pyrimidine always matches with 1 purine, and the way we found out that it works is that A always seems to want to hook up with T, G always wants to hook up with C, and vice versa. And so if you think of them like puzzle pieces, A and T fit together, and C and G fit together, and we're going to, if we haven't already done so, I usually do an activity that shows you how that works. Again, this is just looking at the base itself. You're missing the sugar and the phosphate that are over here. We're just looking at the bases here. Now here's a really good one. So this blue is the sugar, the light blue is the phosphate. Sugar, phosphate, sugar, phosphate. That makes up the sides of this ladder. And in the middle is the bases. And because we call DNA a twisted ladder, it has 2 sides to it there. So wherever you see the A, the other side is a T. Whenever you see a C, the other side's a G. If this is a G here, it's a C over here. They always base pair that way. And there's a weak bond in between them that's called a hydrogen bond. So it's not very hard to break. Alright, so if one side of the ladder said AGC, what would the sides on the other side of the ladder be for DNA? AGC, what would be the other side? And then we mentioned in mitosis, especially during the certain phase, I almost said it, that during one of the stages of cell division, the DNA replicates. It has to make another copy because we're going to make a new cell. Which phase does that happen? And then when you look here, it says in preparation for the cell division, DNA replicates during, like, you have a 1 in 5. The 20% is supposed to say you have a 20% chance of being 1, 2, 3, 4, or 5, obviously. You choose. And the way the DNA replicates is in 3 not very easy steps, but 3 steps that we're going to do. First, there's an enzyme that's a chemical that helps unwind the ladder into almost like a regular ladder instead of a twisted ladder, and that's called DNA helicase. And then there is actually another enzyme that breaks the ladder, almost like taking a saw and cutting the ladder right through the rungs so that you have 2 sides, and then those are called the templates. And then you have literally nucleotides floating around in your cells, and they match up with each of the templates to make 2 new double helices, and we're going to actually do that in class too. Each one has one of the old ones and then one new strand built in it. So take a look at this picture. Here's the original DNA. Here's the sides of the ladder. Here are the bases. This is the blue part. Here's the sugar phosphate. We just don't show it. And then if you look here, there's an enzyme that will then unwind them and separate the bases. And then this yellow is the new sugar phosphate with the new bases. And when you're done, you get two. If you look at them there, CG, CG, TA, TA. You get two identical strands from that base pairing. And that's how your DNA replicates itself during mitosis, or during cell division. The replication occurs very quickly, usually in a matter of a few hours. And usually it unwinds at a certain point and then gets built out from the middle out, like this. But you end up with two brand new, what we call daughter DNAs. And the reason why DNA replication is important is because you need an exact copy of the genetic information, and because you're going to make two new cells, they actually have to have a total of 46 or 23 pairs of chromosomes. So all 23 pairs of your chromosomes, all 46, have to duplicate to like 92 or 46 pairs, and then split so that each cell gets 23 pairs again, diploid. And as you know, that helps preserve the genetic information over time from one organism to their offspring. Hope that makes sense to you guys, and we're going to talk in class and do a lot of practice with this too. Thanks. We'll see you in class.